Hey everybody, hope you had a good long weekend, and we are back to it. This lecture is going to be on carbohydrates, and you have a carbohydrate assignment for today. And I think the quiz we're going to take will be a combination quiz, carbohydrates and lipids, but I'll have to double check on Canvas for that. Alright, so carbohydrates are a third major group of biomolecules. I think at the moment we've talked about one major group. We've talked about the proteins. Now, nucleic acids, lipids, and carbohydrates are the other three. Now, I guess I should say in the order of events for us, this is the second major group. Lipids will be our third, and then nucleic acids the fourth. This diverse group is commonly described as sugars or saccharides, for the Greek from the Greek word for sugar. Now, the simplest of these are called monosaccharides or simple sugars. And an example is glucose. Monosaccharides can be joined together to make larger molecules, such as disaccharides, which contain two sugar monomers, we could say. So on the top here, you can see just a few different monosaccharides. We'll go into some of the naming and the Ds that you see there in a minute. We'll talk about the numbering also. So monosaccharides on top and a disaccharide. This example that I've shown for you guys here is sucrose, which is made up of a D-glucose and a D-fructose. So these two combine together and will make disaccharides. Polysaccharides, chains of many sugar subunits, and these include, and we'll look at, glycogen, cellulose, amylose, amylopectin. And all of these that I just mentioned are actually polymers of glucose, but with different connections, different configurations. Carbohydrates literally means hydrates of carbon, and they follow this kind of formula: CX H2O X. So however many C's you have, will have that many O's. A common sugar that we see often is the glucose formula C6 H12O6. But you can see that if we stick a 6 there, that would be 12 H's. So that's where they get their names from. Okay, not all sugars will have this formula. Typically, there's between 3 and 8 carbons. Deoxyribose, though, for example, is missing an oxygen. It doesn't have as many as a regular sugar would, as a ribose sugar would. So they don't all have to have this formula, but that's where they get their names from. Now, carbohydrates are important in cells as energy sources. They're important as markers of cellular identity. We're going to talk about oligosaccharides on the surface of cells. We're going to talk about carbohydrates as structural components, such as cellulose in plants. And they're also constituents of nucleotides, of the fourth, the fourth group of biomolecule. They make up the sugar in the sugar base of our DNA backbones. Okay, so we're going to start talking about monosaccharides first, and the most common of these monosaccharides is going to be, or the most common are going to include, glucose, but also fructose, galactose, ribose, mannose, and actually I probably should have stayed on this slide because those are them. Now, all of these sugars, but one, exist as an aldehyde. Now, if you remember your organic chemistry, the aldehyde is the H directly attached to a carbonyl carbon. Now, all of these except for fructose. Fructose and some other lesser known sugars are ketones, and they have a ketone group. Again, a ketone group here on fructose. And so let's talk a little bit about the nomenclature here. So by convention, letters OSE at the end of a name flag that it's a sugar. So glucose, galactose, sucrose, right? They have all these OSEs at the end, OSEs. Now, there's other descriptive nomenclature, including a prefix, and this will tell us essentially how many carbons there are. So in 3-carbon sugar, we would call that tri. And so here you can see I'm already starting to mix the different names. So we've got aldehydes that are going to be aldo. And we actually break up our sugars into two groups. We call them the ketoses, or the the ketone containing groups, or the aldoses, the aldehyde containing groups. And then we also will give it the OSE ending, right? That signifies it's a sugar, and then the different numbers of carbon are designated by tri, tetra, pent, hex, and so on. So we use the prefixes to tell how many carbons there are. Now, they're also given sometimes common names. Glucose, for example, is an aldohexose, but that doesn't describe exactly which aldohexose it is. There are lots of aldohexoses. Glucose, galactose, mannose, fructose, those are all aldohexoses. So they're often named with either common names or a chemical name. All right, let's see. So, oops, those are the monosaccharides. I guess all of that was on here. I could have shown you that. Oops, jump back real quick. Aldotriose example, glyceraldehyde. We're going to talk about that molecule in glucose metabolism. Okay, so monosaccharides will either contain the ketone or the aldehyde group. Let's talk a little bit about enantiomers. Now, let's see. We need to back up a second and talk about chiral carbons. Just to remind you guys what a chiral carbon is. We talked a little bit about it with the amino acids, but just a quick reminder, a chiral carbon is a carbon that has 4 unique groups attached to it. And we would designate each chiral carbon as being either L or D, depending on it's variation of the way that you would do it in organic chemistry, assigning S or R, depending on which type of rotation we see, which kind of polarization of light as it passes through the sample. Chiral molecules will affect light that way. We could also give importance to different groups, all the different 4 groups, and then arrange them in order from most important to, or least important to most important, and or most, I forget the word exactly that we used in organic chemistry for that. And depending on whether that order is clockwise or counterclockwise, they get L or D. We're not going into that here. So sugars may have asymmetric carbons, and so they can even have multiple asymmetric carbons. Asymmetric carbons here being chiral. So 2 sugars can have the same chemical formula, like the aldoses we listed off a few minutes ago. They can have the same number of carbons, but they can only differ in the stereochemical orientation of their carbons. And we refer to these as diastereomers. So, for example, glucose and galactose, and mannose, for that matter. Let's see, do I have those here? This is just 2 different glucoses. Let me go back to glucose and galactose for this description. So glucose and galactose, we're talking about this guy and this guy. So if you look at them here, you can see that they are very, very similar. In fact, the only difference they have is carbon number 4. Carbon number 4 has the position of these OH groups flip-flopped. So these guys are diastereomers. Glucose and galactose and mannose. Where's mannose? Mannose is here. Mannose is actually the same as glucose with the exception of carbon number 2. The OH on carbon number 2 is in a different position. And so these guys are all chemically distinct from each other in the position of those hydroxyls, and so they are referred to as diastereomers of each other. So just different arrangements in space. We have something called epimers. Let's talk about epimers. Now, epimers are like the 2 different glucoses that I was showing you here. Oops. Here we go. Okay, so if 2 sugars are identical, except for having one hydroxyl configured differently, kind of like glucose and galactose, they're diastereomers, known as epimers. If the configuration of the hydroxyls of one sugar is exactly the opposite of the configuration in another, then they're mirror images of each other, and these are called enantiomers. So let me back up. Epimers are molecules that have the same formula but different orientations in space. Epimers come in two flavors. They can come in diastereomers. Diastereomers are the ones like glucose and galactose that are just different yeah, from on one carbon from each other. Now, not every diastereomer is a mirror image of if it's other molecule. So diastereomers, galactose and glucose, they're not mirror images of each other. But these two right here, these are enantiomers. These are mirror images of each other. So of course they have the same formula, but different orientations in space. Enantiomers are when the sugars are exactly the opposite. So here you can see OH of carbon 2 is on the right, OH of carbon 2 is on the left in the other molecule. Now, excuse me. Um, so, so due to the way sugars are named, L-glucose is the mirror image of D-glucose, and vice versa. Or same for like glucose, galactose or fructose. The L version is going to be the mirror image of the D version. That's just that's how they name these things. Let's see. Looking at my notes here real quick. I don't want to miss anything about these sugars. Let's talk about how we actually decide whether it's going to be D or L for us, for sugars, and that's actually really easy. So for sugars, the way that we decide is that we look at there's a couple of books that say it a different way. The last chiral carbon from, sorry, let me back up one more second. When we talk about sugars, we often show them in these Fisher projections. Now, what we're looking at right now, what we've been looking at, are Fisher projections. They're ways of representing a molecule in completely flat paper like this. The bonds, all of the sideways bonds are in the same orientation. All of the up and down bonds are in the same orientation. And so this is just the way that they represent them. I forget if the sideways ones are pointing away from us and the up and down bonds are pointing at us. It's something like that. So we always put in Fisher projections the most oxidized carbon at the top. So you're going to get your ketones and your aldehyde groups at the top. Now, the last chiral carbon in this molecule is going to be the last carbon that has 4 unique groups on it. And so it's obviously not this carbon. This carbon doesn't have 4 unique groups, but it is the most oxidized carbon, and it always goes at the top. Here's our first chiral carbon, second chiral carbon, third chiral carbon, fourth chiral carbon, and then this carbon right here is not chiral because it has 2 hydrogens on it. So the last chiral carbon, or the furthest chiral carbon from the top, is the one that we use to determine L or D. If the OH is on the right, then it's D. If the OH is on the left, then it's L. And so you can see here what that looks like. So here are a couple of examples of other D sugars. Now, our bodies tend to use the D sugars and not the L sugars. This is a little bit of a flip-flop from the amino acids, right, where we use L's and not D's. So again, in this sugar here, which would be an aldose, a 1, 2, 3, 4 aldotetrose, right, with a specific name 3-ose. You don't need to know the common names of these sugars, but we know it's D because of the position of the OH group. So anyways, that's what that's about. Okay, so let's talk about what sugars do. Now, sugars with 5 and 6 carbons will readily, what's the word I'm looking for? They'll readily cyclize. Now, when they do, a new asymmetric carbon is created, one that wasn't in the previous sugar when they were in their straight chain forms. This carbon has a special name. It's called the anomeric carbon. I'll show you a couple pictures in a minute. Now, like the other asymmetric carbons in the sugar, it can have the hydroxyl group in 2 positions. Now, these positions are referred to as alpha and beta, and I'm going to show you, again, some examples here in a second. And so actually let's just take a second to look at the reaction of the cyclization, and then we'll go on to the anomers. So here you can see this is a 5-carbon sugar. So again, 5-carbon and 6-carbon sugars are capable of doing this. Now the reaction, if you remember some of your organic chemistry reactions, are what we call a hemiacetal and an acetal formation. So a hemiacetal is a reaction between a carbonyl and an alcohol. So a hydroxyl group on an alcohol will react with a carbonyl group on either an aldehyde or a ketone, and they will react to form what we call a hemiacetal. Now, a hemiacetal is what we're looking at right here. Hemiacetal has a hydroxyl group. It's still connected to an oxygen. The difference is going to be that when we get a full acetal, there won't be a hydrogen anymore. So that's how you kind of differentiate a hemiacetal from an acetal. Now, the reason I'm even talking about acetals is because when we form disaccharides and polysaccharides, this will be the acetal formation. Okay, but hemiacetal is what we form with the cyclization of a sugar, and you can see here that it's the carbon number 4 that's going to attack the carbonyl carbon here. I'm sorry, the oxygen on the carbon number 4. The ring sort of folds over so they can get close together. Lone pairs on the oxygen are going to attack this, causing this bond to jump up. And then we get some hydrogen sort of rearranging here. This is going to grab this hydrogen. This one's going to drop its electrons back down, and we get our hemiacetal. Now, when that happens, the position of this OH group is sort of random. It could be in the up position, which we'll call beta, or it could be in the position it's shown now, and we'll call that alpha. And I think, oops, that's some text here. Here we go. So here, another example. This one is actually just showing a 5-membered ring, like we were just looking at, but it can actually fold into a few different types of sugar rings. So we'll talk about that second. Let's see. What I wanted to show you is down here. So this is beta fructose and beta glucose. Nope, that's not what I want to show you. Here we go. This is what I want to show you. So this is glucose. This is also another name for glucose. This is not the common name. This is glucopyranose is what this is called. Pyranose describes the 6-membered ring that we're seeing here. 6 members being 5 carbons and an oxygen. But here we can see alpha D, glucopyranose, because the OH on the anomeric carbon. So let's kind of talk about how we know where the anomeric carbon is. The anomeric carbon is usually going to be shown right here. It's going to be the number 1 carbon, if you can think about it that way. The number 1 carbon. If we go back to the ring, or even here, we can kind of see that. The number 1 carbon, of course, in fructose, it's not the number 1 carbon, it's the number 2. It's whatever carbon has the carbonyl group on it. So if we're going back to these pictures, if we're going back to these previous slides, these are the number 1 carbons right here. Okay, and some oxygen on a further down carbon is going to come in here and react with this and cause this carbonyl to turn into a hydroxyl. And when the hydroxyl's up, we call it beta. When it's down, we call it alpha. And that's what we're getting at here. So alpha is when it's down, and sometimes that can just be up when this ring closes. Now, these convert back and forth into each other. This alpha and beta, this ring structure is constantly opening and closing, opening and closing, opening and closing. And so these things do flip back and forth. Now, the beta form is a little more stable in solution, and so we actually do see that one a little bit more often. Okay, I want to go back to this real quick. So fructose is one of these that can actually form 2 different ring structures. Now, when it forms a pyranose or six-membered ring, it would look like this. When it forms a furanose or five-membered ring, which is what fructose tends to do, we see it like this. So the reason that it can sometimes, form different ring structures will depend on which carbon it looks like here is actually doing the attacking. In the furanose ring, it's going to be carbon number 4 that attacks the anomeric carbon. In the pyranose ring, it looks like it's carbon number 5 that does it. So there's some flexibility there. Let's see. Now, why am I showing you these guys down here? The alpha D-glucose and beta D-glucose. I'm sorry, the two beta D-glucoses. There was a reason for that. I guess just to show you, again, the two rings, the furanose and the pyranose ring. So pyranose, six-membered, furanose, five-membered. Okay, and I mentioned they flip back and forth in solution. Most sugars can form furanose and pyranose structures, by the way. Let's talk a little bit about boat and chair conformations. So, orbitals of carbon prefer to be in tetrahedral conformations. So this means that the bonds between carbon in a ring do not lie flat, though we often show them sometimes as flat rings. So indeed, these rings are sort of like puckered. And they do this to try to relieve any sort of, to be in a tetrahedral conformation, but also they tend to flip around based on where the groups are pointing, because those can give rise to kind of what we call steric hindrance. So some of these forms resemble what we call boat structures, and others resemble chair or, I guess, kind of like envelope structures. The stablest and the most abundant of these forms have all the hydroxyls in the equatorial positions, and this basically relieves any kind of steric hindrance. And so you can see here in the boat conformation on the left, we've got some, let's see, I'm just going to circle or like point out all the equatorial and axial positions here. But it looks like when you look at the equatorial positions, I think those are the only ones actually being shown here. If you look at all the equatorial positions of the OHs, there's a little bit of some grouping, like a little bit of crowding maybe on this one. But if you look at the chair conformation, they seem to be all sort of pointed in their own directions and kind of, not getting in each other's way. And so chair tends to be the more stable of these two orientations. I mean, you can even see that here. This group would be pointed up and this one down, where these two would be on the same side. So just a little bit there on the kind of form that we're going to see the sugars in. All right, let's see. I want to get into a topic about some modified monosaccharides. That means we need to talk a little bit about their bonding. Okay, let's talk about glycosides. So glycosides are bonds between sugars. And you're going to see some bonds between sugars actually right here. So we've got this bond right here is a bond between two sugars. This is lactose, the disaccharide, and then there's another bond between two sugars right here. I guess we could extend that to be like all of this is the bond. Sucrose. So we'll talk a little bit about those specifically. All right. So let's talk about, before we look at how sugars connect together, because those structures can be a little complicated, let's just look at the reaction using a smaller molecule here. So I've got what is this, ethanol shown here. So the reaction that takes place in glycosides takes place between the anomeric carbon, so this carbon right here, and a hydroxyl group on an incoming sugar. Now, the hydroxyl group is usually going to be, like this hydroxyl on the next sugar. Do you know what I mean? And they're going to connect together in that fashion. Or this hydroxyl on carbon number 6 will come in and attach. But we'll show the reaction here first. So we've got the hemiacetal here, this carbon connected to an ether. It's basically when you have two ethers. Here's one ether bond, and then we've got a hydroxyl. At the end of this, we're going to have an acetal. So this guy here has one ether bond in that direction, right? This carbon. One ether bond, one hydrogen, another ether bond, and then just the rest of this carbon bonds. So that's the acetal right here, these two ethers. I misspoke when I said we'd lose the hydrogen earlier. We're going to form two ether bonds to the same carbon. So these are glycosidic bonds, these are glycosides. Now, when a carbon undergoes a glycoside reaction or becomes a glycoside, it forms a linkage, right? It locks the anomer. So for one, that locks you in place. So this is the alpha version of this sugar. Well, now we have an alpha glycosidic bond here because the O is down. The O is down, down is alpha. Okay, so it also stops the sugar from being able to be a reducing sugar. So this now becomes a non-reducing sugar. So let's talk a little bit about what that means. So I'll touch up on oxidation reduction in a second. Let's look at just how we get reducing sugar or non-reducing sugar. So when you have an anomeric carbon that is unbound or free, you got to remember that the ring opens and closes all the time. Remember, it opens and closes, and this OH goes from being beta to alpha, beta to alpha. Whenever it's open, okay, when the ring opens, it's not a hemiacetal anymore. It's a aldehyde on the end of that carbon, on the end of that sugar. And aldehydes can be reduced. I'm sorry, that's a lie. Aldehydes can be oxidized. They can be oxidized. Well, actually, they can be reduced as well. And so I misspoke when I was saying that they can't be reduced. They're right in the middle. They're right in the middle. If you don't remember, alcohols of the primary variety become aldehydes when you oxidize them. Secondary alcohols become ketones when you oxidize them. Aldehydes can be oxidized even further into carboxylic acids. Okay? So an aldehyde or a ketone at the moment is kind of right in the middle. The aldehyde's in the middle. So an aldehyde can be reduced back into an alcohol, right? Or it can be oxidized into a carboxylic acid. And those that can be oxidized into carboxylic acids are what we call reducing sugars. Remember, the thing that gets oxidized is the reducing agent. The thing that gets reduced is the oxidizing agent. So when we say that a sugar can be a reducing sugar, it means it gets oxidized or can be oxidized. Now, just looking at these two sugars, I want to point out that the reason that this one is a reducing sugar is because it has a free anomeric carbon. And that anomeric carbon can undergo opening and closing of this ring, cyclization and uncyclization, right? And when it's not cyclized, it's got an aldehyde group which can then be oxidized. On this non-reducing sugar, and by the way, a ketone is also a non-reducing sugar, like fructose. Well, fructose is a reducing sugar, but only because it does something called mutarotation, where it actually, or isomerization, where it can actually turn into glucose in our bodies. And so, technically, though, we wouldn't say that glucose, that fructose, which is a ketose, is a reducing sugar. But we can say that it does end up being a reducing sugar once it turns into glucose. But because it has a ketone group on it, it doesn't oxidize. Therefore, it can't be a reducing agent. And since both of these anomeric carbons in this sucrose, this is fructose, by the way, we've been talking about fructose, both of these anomeric carbons are bound up in a glycosidic bond, therefore they can't open and close anymore. Therefore there's no aldehyde group showing or no ketone group showing in the case of being, oxidizing sugars, and so they can't participate in these reactions. So that's how you kind of know if you're a reducing sugar or um, or I guess we don't really call it. We don't call the other version oxidizing sugars. We call reducing sugars. Well, the products of reducing sugars are what we call sugar acids, and then um, when we actually reduce a sugar, okay, don't get caught up in the terminology here and get confused. A reducing sugar does not get reduced. A reducing sugar is oxidized. Aldehydes oxidize into carboxylic acids. So a reducing sugar becomes a sugar acid. But if we reduce a sugar, we get something called sugar alcohols. And we will talk about all of these right now. Actually, I think maybe we're going to do some naming. Let's see. Oh, no, that's just describing the bonds. We'll come back to that. So on the topic of the reducing sugars and the non-reducing sugars, I just wanted to say one last thing before we... Actually, I do have some slides in a minute that'll be a little more informative. So we will move on. We'll move on real quick and talk about the nomenclature of the bond. So as far as the bonding goes, and we can throw the words up here. So sucrose would be, you can see down here the name alpha D glucopyranosyl 1,2 beta D fructofruinoside. That's a big name. Another way that we would do this is we'd say that this is D glucose. Actually, we would say this is alpha D glucose. Glucopyranosyl is the formal name for glucose. We could say that this is, we look at the position of the OH. The OH isn't even shown here. Actually, my bad. We use the position of these groups on the sucroses. No, that's another lie. I'm sorry. I'm looking at this sugar backwards because they turned it around so that we could connect it. So the number one carbon is actually right here. And the number one carbon has the OH group. Looks like it's got it down. But again, that might be just because of the way that they show the sugar here. Let's not look at this guy. Let's look at this guy. This is how they usually show sucrose, and it makes the bond naming a lot easier. So our alpha glucose is alpha because the OH is down. That participates in this glycosidic link. And in the so this is actually the number 2 carbon, right? Because that was where the carbonyl group was in fructose. The OH was up, so that was beta. So we would call this an alpha beta 1,2 glycosidic link between D-glucose and D-fructose. Alpha beta 1,2. You can kind of see that here. Alpha beta 1,2. So when we talk about linkages, we're going to talk about the important ones. For us, important ones, well, in this disaccharide business, alpha beta 1,2 is important. In some of these other disaccharides like lactose and mannose, they're usually alpha beta, alpha 1,2. Alpha 1,2 is reserved for sucrose because those are the 2 anomeric carbons. In most other disaccharides, we don't actually have to worry about the 2 different anomeric carbons. There's usually only 1 anomeric carbon. So they say something like alpha 1,4. This would be the linkage in our mannose or in our amylose or in our amylopectin. We're going to have alpha 1,6s, and we'll talk about beta 1,4. That's what cellulose has. So when we say things like that, we're talking about whether the anomeric carbon is, the hydroxyl was up or down, alpha or beta, and then which 2 carbons are linked. The number 1 carbon to the 4, 1 to the 6, and so on. Okay. Modified sugars. So I've got a few different modified sugars here for you guys to look at. Now, there's different reasons why modification of sugars happens. One common type of modification includes oxidation or reduction, which we're going to go over a little more, phosphorylation. We saw that with proteins. Substitution of an amine or an acetylamine in place of a hydroxyl. So amino groups or some kind of amino, like you can see here. So galactosamine is a part of that, or acetylglucosamine. So this whole little bit, acetylamine was added. Phosphate, right? This is a oxidation of a sugar to the acid form of it, sugar acid. Okay, there's no sugar alcohol shown here, but anyways, so the ones that affect the anomeric hydroxyl group make up glycosides. So if you've heard of glycosides, that's where the name kind of comes from. These types of those bonds that we just looked at. And there are also modifications that don't affect the anomeric hydroxyl carbon, or the anomeric the anomeric hydroxyl carbon. So those would not be glycosides. They wouldn't have a glycosidic linkage. But again, they would still be various. They would still be forms of a sugar, modified sugars. Okay, so this is an example of some oxidation and reduction that I wanted to talk about. So we have a few different ways of oxidizing sugars, and one of the most common ways is to use something called a Benedict solution, which actually uses copper 2 plus ions to get reduced. I think the copper 2 plus gets reduced into copper 1 plus, which changes a color, right? Copper 1 plus produces an orange precipitate in oxygen, and so we can tell if the sugar we're testing is actually a reducing sugar or not. So this is a Benedict solution with some sugar added. We need a little bit of heat in there, and in the event that the copper actually gets reduced, that means the sugar got oxidized. And when a sugar gets oxidized, let's see if I can show you that. We get this guy. So the one of the so this certain pH, right? This would be an acid. This would be a sugar acid. So C double bond O, OH, but then of course it can lose that proton and be negatively charged. So Benedict's test, also used in the medical industry to test urine to see if there's any sugar in there. They use these Benedict solutions for that. Now, the aldehyde group on a sugar is actually very susceptible to oxidation, whereas ketones are less so. But it can be oxidized if it's like fructose, which can, like I mentioned before, it can change into an aldose. Most monosaccharides are reducing sugars. This includes all the common ones: galactose, glucose, fructose, ribose, xylose, mannose, to name a few. Some disaccharides, such as lactose and maltose, are reducing sugars since at least one of the anomeric carbons is free. And so that part of the sugar can linearize and yield an aldose. Sucrose, we mentioned, has no anomeric carbons free, so they can't participate in these reactions and are not reducing sugars. Oxidation and reduction of sugars occurs in the cells. We do see that sometimes. Phosphorylation of sugars also occurs during metabolism. Glucose 6-phosphate, for example, is an example of that. Glucuronic acid, also an example of a oxidized sugar. This glucuronic acid is actually a common. That's what I'm showing you right here. Common precursor to, or common, constituent of glycosaminoglycans, proteoglycans. We're going to talk about some of those. Okay, and then a sugar alcohol. Sugar alcohol is what I'm showing you right here. Sugar alcohol is from the reduction of an aldose or a ketose, usually from a hydrogenation process. And these are often used as thickeners of food or artificial sweeteners. They do stimulate the sweet receptors on the tongue. This one down here is actually sucralose. It's one of the more common sugar alcohols. Glyceraldehydes was also one, xylose, sorbitol. Anyways, these have a sweetness between, 0.4 and 1 times as sweet as sucrose. Less calories because they don't, they're not absorbed by the intestines very well. They often used to mask the aftertaste of the other artificial sweeteners, sugar alcohols, that is.
Most of these actually, even when they dissolve in your mouth, the compound, due to endothermic processes, result in it like a cooling sensation, and so usually they're pretty pleasant in that sense. Artificial sweeteners are compounds that stimulate the taste receptors, but aren't metabolized for energy at the same efficiency as, let's say, sugar is. These compounds are usually many times sweeter than table sugar or sucrose on a weight-by-weight basis, um, and are usually referred to as intensely sweet. Most of these artificial sweeteners are not carbohydrates, but are able to stimulate the same sweet receptors. There's a few that you would probably heard of: stevia, aspartame, that's found in equal, saccharin, sweet and low. Sucralose is often it's a sugar alcohol. Actually, that's not true. Sucralose is different than sorbitol. Sorbitol is a sugar alcohol of glucose that's sometimes used as an artificial sweetener as well. Advantame, I think, is another one. Anyway, saccharin. Oh yeah, it's on there. Disaccharides are made up of two monosaccharides, the most common being sucrose, which is what we've looked at, glucose and fructose together, lactose being galactose and glucose, and maltose, which is just made up of two glucoses. They all contain at least one glycosidic link, and we talked about a little bit about how those are named. Okay, oligosaccharides. Now, oligosaccharides are comprised of a few, oligo being the word for a few, usually 3 to 9 sugar residues. Often, but not always, contain modified sugars. And unlike other saccharides or oligosaccharides, they're typically not found unattached to other cellular structures. These things are usually bound. So other saccharides can be floating around, obviously being digested, but these guys, oligosaccharides, are usually bound to structures. Oligosaccharides, for example, are bound to sphingolipids, which make up like cerebrosides and gangliosides, which contain some sugars, or proteins, making glycoproteins. We'll look at a few of those. Um, Oligosaccharides in membrane glycoproteins actually play important roles in cellular identity and recognition. Patterns of oligosaccharides that are displayed on the extracellular face of the plasma membrane kind of act like a barcode that identifies specific cell types. The immune system recognizes these identities tags as, body, and any foreign structures or foreign oligosaccharide structures trigger the immune system to attack them. Um, this is a good thing, but it's also a bad thing. Let's say if you get an organ transplant, right, from one individual to another, these markers might be different, and then so of course your body may not recognize them. We're going to talk about a little bit more of organelle targeting. So oligosaccharides that are attached to proteins can also determine their cellular destination. So we know that proteins sometimes get made in one region and transported to another, and so oligosaccharides play a role in, I wanted to say, like a mail carrier role. They have the address for that protein sort of written on it. Improper, and we call this glycosylation. Improper glycosylation can actually lead to the failure of proteins to reach the correct cellular compartment, let's say. There's a disease called, it's called inclusion cell disease, I-cell disease for short. It comes from a defective phosphotransferase in the Golgi apparatus. This is where the packaging of this protein happens. Enzyme normally catalyzes the addition of a phosphate to a mannose sugar that's attached to the protein destined for the lysosome. And in the absence of a functioning enzyme, the unphosphorylated glycoprotein never makes it to the lysosome, and instead gets exported out of the cell. It accumulates in the blood, excreted in the urine, and so people with I-cell disease suffer developmental delays, abnormal skeletal development, restricted joint movement, all because This protein isn't getting phosphorylated properly. And of course, the protein being the glycoprotein part of it, the mannose residue. Sugars are commonly attached to proteins in a process called glycosylation, and there are 5 major classes of glycosylated products. There are N-linked glycans. They're called glycans if multiple carbohydrates are attached via glycosidic bonds. So there are N-linked glycans, there are O-linked glycans, and we're going to take a look at those, these two right here, and maybe give you a little more info on them. There are phosphoglycans, there's glypiation, sorry, and there's C-linked glycans. Now they all differ in how they link, okay, but they have several similar functions. Some of them have to do with the folding properly or the stability of a properly folded protein. Some serve as the cellular identifiers, like blood types, for example. Different glycosylation of blood cell membrane proteins give rise to the different blood groups. Can also be important in cell adhesion, cells sticking together, and in the immune system. So a little bit about the N-linked glycosylation. So this process occurs in the endoplasmic reticulum. It involves building a part of the glycosyl chain on a molecule called dolicholpyrophosphate. I don't think I have a dolicholpyrophosphate picture for you, but and then another feature of it is that it has a common core structure of carbohydrate residues that are linked to proteins. I have a picture of that. So I'm referring to this sort of little structural backbone. You can see it in all of these N-linked, with the exception of this guy, having a modification of it. But that little, repeating unit of these two sugars, again, is common across animal kingdoms. It's called N-linked glycosylation. N-linked glycosylation, if we go a little further on it, all of these are post-translational additions to oligosaccharides once they get attached onto proteins anyways. So there's some post-modification that happens. The formation of these glycosidic bonds on proteins requires that there's a free OH or a free amino groups. And so often we're going to see like serine residues or aspartic acid residues, asparagine, used because the amino acid, so this is the amino acid, has that free group that can make a bond with the anomeric carbon. Again, on our serine residue, this would have been an OH right here, and we know that OHs are reactive with our anomeric carbons. So for glycosylation, for O-linked glycosylation, we need serenines and threonines and things that have OHs. For the N-linked glycosylation, we need asparagines and other amino acids that are going to have nitrogens on them. Okay, and so I was talking about N-linked glycosylation and showing you a little example here. We can talk about O also very quickly. O-linked is also initiated in the Golgi apparatus, so some stuff starts in the endoplasmic reticulum, but primary processes started in the Golgi. One of the threonine, serine, or tyrosine residues gets modified. I don't think they've really figured out the direct order in which things happen here, but often these O-linked glycosylations start with a specific sugar called N-acetylgalactosamine. We see N-acetylgalactosamine often in bacterial membranes. Let's see, is there anything else that's important to talk about with these guys? I don't want to get too into the details of the types of O-type or N-type glycosylation at the moment. Okay, I think we can safely move on. Oh, I did have this image that I wanted to talk about. So, in regard to are they important and how do they help to stabilize proteins? So this is a recombinant protein, glycoprotein. Um, Erythropoietin, I believe, is the real original protein. This is a recombinant. This one actually does illustrate the importance of the glycosylation to function of protein. So, a mutation of an asparagine to a glycine or to a glutamine, sorry. So just a change of an amino acid even resulted in the drop of the secretion levels in this and loss of structural stability. So changing the amino acid that would be linking these oligosaccharides, clearly that means the oligosaccharide then isn't there because it's not linking, has an effect. In a couple places it was drop in secretion levels, which means it might not be being packaged up and transported the way it's supposed to be, but also loss of structural stability. So some interaction keeping the molecule in its proper folding. Okay, so importance of oligosaccharides in our glycoproteins. All right, switching gears, let's talk a little bit more about um, polysaccharides. And so what I'm showing you here is the structure of amylose. And so polysaccharides are long polymers of sugar residues that can be thousands of units long. These are found free, so not attached to other molecules or bound up in cellular structures like proteins. Polysaccharides are just kind of floating around. Some of them are homopolymers, meaning that they can contain only one kind of sugar. So like glycogen, cellulose, amylose, callose, all of those are homopolymers of glucose. And then chitin is a homopolymer of N-acetyl glucosamine. We saw that with our disaccharides even, that they're heteropolymers often. They don't have the same monomer over and over, like a glucose and a galactose, or a glucose and a fructose. And we do have some of those heteropolymers as well, not listed here. Glycosaminoglycans, hemicellulose, for example, are two of them. Polysaccharides function in energy storage. They can function in structural enhancement, kind of like chitin and cellulose. They can have a lubrication role, like hyaluronic acid does. Um, and so we'll talk a little bit about some of these different polymers. So let's see. So amylose. Let's talk about amylose first. So amylose is the picture I've got here shown for you. This is the simplest structure of any of the nutritional polysaccharides. It's just made up of glucoses linked by alpha 1,4 glycosidic bonds. The term starch that we usually use is actually a mixture of, so starch, right? I think I'm going to have a picture right here in a second. Starch, being the stuff that you get from potatoes and corn and bread and all that stuff. This is a mixture of amylose and amylopectin. Amylose is an unbranched chain, where amylopectin is branched chain. Amylopectin contains an additional type of linkage. So amylopectin is shown up top in the structure. So you can see amylose would just be alpha 1,4 linkages between all of the glucoses. Amylopectin is branched, and so we have an additional alpha 1,6 linkage. It's alpha for the anomeric carbon, the OH being down, and number 1 carbon being the anomeric carbon, and then it's attaching to the number 6 carbon on the neighboring sugar. So it's like this is where it branches off, but you can see that two different types of branching. We have the straight chain branching, and then, or the straight chain glycosides, and then the branching glycosides. So again, usually a mixture of the two. Now, different plants, different vegetables, they have different ratios of amylose to amylopectin. Amylose is insoluble in water. It's harder to digest than amylopectin. Let's see. By actually mixing these two together, having amylopectin and amylose together facilitates its water solubility. Let's see, and also its digestion. Amylose is produced in plants for energy storage, and since Plants don't have rapidly changing demands for glucose. No muscular contractions. As an example, its compact structure and slow breakdown are consistent with plant needs. So amylopectin looks a lot like glycogen. I thought I had a picture. I don't have a picture. So I do have a muscle man here. Now glycogen is just like amylopectin, with the exception that it has more branches. So it's even more branched. So more complicated homopolymer of glucose is amylopectin in plants from amylose, and then glycogen in animals. So both glycogen and amylopectin have long chains, alpha 1,4s. Every 25 to 30 residues or so, they have a branch, an alpha 1,6 branch. Um, for amylopectin, for glycogen, that might be every 8 to 12 residues. So way more branching. Now, more branching means more water solubility, and and providing more ends of the polymer. So having more free ends means more sugar can be broken down and freed up when needed. So a faster release of sugar than a plant would get with less branching ends and less branching. All right. We're going to talk about carbohydrates and energy breakdown. So that's about all we need to say about it right now. So storage for energy vary to the needs. Plants have one long big chain of amylose, you know, two free ends, one free end, really, if you think about the side. We can only digest it, one molecule at a time. Whereas branch structures, enzymes can be cleaving off the outside glucose, and you get all of these at once. More branches means more glucose is all at once. So that's all we need to say about that. Let's talk a little bit about structural polysaccharides. Um, the one I'm showing you here is actually cellulose. So this is cellulose. It's got a beta, right? So this is a 1,4 linkage, and it's beta because the OH is up. Okay, cellulose. This is also a polymer of glucose with a different linkage. It's an important structural component of plants and fungal cells. Notably, most ruminant animals are unable to digest this polymer. We don't contain in our bodies the enzyme called cellulase. So, cattle, they do have bacterium in their one of their stomachs or in their the digestive system that does have the enzyme, and they're able to get glucose from plants. We are not. There's another group of polysaccharides in plants called hemicelluloses. These encompass several branched heteropolymers, mostly D-pentose sugars. These are 5-carbon sugars, or 5-carbon ring sugars. 5-membered rings. Sorry, 5-membered ring sugars. We'll look at some hexoses and so again, branched heteropolymers, D-pentose sugars with some hexoses and some L sugars. So pentoses, being 5-carbon sugars. No mention of the ring, whether it's furanose or pyranose. I was confusing myself with that. Let's see. As far as monomer sugars go in these polysaccharides, besides glucose, we can have polysaccharides of xylose, of mannose, of galactose, of arabinose. I think xylose, though, is the one that's most commonly found, and so that's why I've got it here. This is xylose, other than glucose, that is. There's chitin. I don't know if you guys know about chitin. Oops, I don't know what I'm trying to show here by blocking it with my pen markings. So let's talk about chitin. I think I have a good structure of actually what I'm showing you here is some more cellulose. So cellulose stabilized by hydrogen bonding between sugar residues, in addition to the linkage there that we can't digest, makes this a really good fiber. It makes it really good at having some mechanical strength, some sturdiness. And so cellulose is, of course, the rigidity that we see that plants get. So cellulose fibers are high in strength. Fiber in general indigestible by humans. Okay. So I wanted to talk now about chitin. So chitin is a N-acetylglucosamine monomer, uh, monomer polymer. So this is N-acetylglucosamine. That's the monomer. The linkages are beta 1,4 glycosidic linkages. Um, like cellulose, this is enhanced by hydrogen bonding between adjacent strands. Um, and so this is also non-digestible by humans. Chitin, you can see in that picture there on the wing. This is of a sap beetle. Again, structural polysaccharide. We also see chitin, main component of cell walls of some fungi. Also, exoskeletons of arthropods, insects, and stuff like that. The structure of chitin was actually solved in 1928 or 1929. It was really one of the first structures that was sort of investigated. And like cellulose, except for the acetylamine group replacing the hydroxyl on position 2, it would be almost identical to cellulose. So acetylamine group replacing the hydroxyl group. It's basically just a modified cellulose with that additional little structure here. N-acetylglucosamine, beta 1,4. I mean, just like cellulose. And so that really allows for it to have that same sort of bonding pattern, hydrogen bonding pattern. I mean, for stability. Pectins. We can talk about pectins real quick. So this is a powdered form of a pectin. Let me think of what this is a D-galacturonic acid. You don't need to know the name of that, but that's the structure being shown here. So another group of the structural polysaccharides are the pectins. I should write that in case you can't hear what I'm saying. Pectins. These compounds are present in most primary plant cells, abundant in non-woody parts of like terrestrial plants. Rich in galacturonic acid, so there's the structure for you. Alpha 1,4 linkages, no branches. It's used commercially as a gelling agent in jams and jellies, pectin, as well as a stabilizer in fruit juices and milk drinks. There are some health concerns with pectin. You could get reduced blood cholesterol levels because you drink pectin. If you eat pectin too much, it binds your cholesterol, and so you have less in your blood. It also increases the viscosity of the intestinal tract, reducing absorption of cholesterol from food. Anyways, but pectin is a structural polysaccharide. We use it for various cooking things, but let's talk about lectins. So this is a lectin. Now lectins are not carbohydrates, but proteins that specifically bind to carbohydrate molecules. We talked about how oligosaccharides are used as markers for identification. Well, yeah, but something has to bind to them and recognize them, and so lectins are those that do this job. So carbohydrate molecules that are found in animals and plants attached to proteins are called phytohemagglutins. And so there are lectins that have carbohydrates attached to, or that bind certain carbohydrate groups are called phytohemagglutins. In plants, in our body we call them lectins. In plants we call them phytohemagglutins. And I've got a picture of one. This is a phytohemagglutinin right here. Or just hemagglutinin, I think is the name of this, but phyto if it's in plants. It's very specific for certain sugars. So this one that we're looking at here functions in cellular and molecular recognition, as well as cell adhesion. We said cells touching each other, sticking together. One lectin recognizes hydrolytic enzymes that contain, let's say, mannose 6-phosphate, and then targets them to be delivered to the lysosomes. We talked about that eye cell disease. That was a different recognition that was of an enzyme that put a phosphate on, but similar sort of binding recognition or carbohydrate recognition. the innate immune system, our mannose binding lectins actually help defend against invading microbes, because these microbes have these mannose sugars on their outer, protein membranes, or on their outer membrane proteins, so that the backwards order. Other lectins have roles in inflammation and autoimmune disorders. Some viruses and bacteria use lectins actually to recognize and bind specific carbohydrate residues on the surface of our cells. So like the flu virus carries a lectin known as hemagglutinin. That's the one that I'm showing here. Phytohemagglutinins are in plants. Hemagglutinin, specifically in this picture, is the one from a virus that binds to sialic acid and essentially needs that interaction to get into the cell on the target cell. Once it binds to the sialic acid on the outside of that cell, the target cell, endocytosis then brings in the hemagglutinin, or brings in the particle. Hemagglutinin itself gets like cleaved off, and then once the virus is inside, well, we'll talk about what happens with viruses when they get inside a little later. Let's see. I think that might be the end of the chapter, or the end of this lecture here. There are some other polysaccharides that are worth mentioning. Agar, or agarose, if you've ever done some microbiology work with gels and bacteria, we use that polysaccharide to grow bacteria on it. It's a polymer of D-galactose and 3,6-anhydro-L-galactopyranose, or something like that. Don't quote me. Let's see. Agar gel. There's other types of gels that we use as well. Glycosaminoglycans for, what is the word? Electrophoresis and stuff. Heparin. We talk about heparin's a modified polysaccharide. Sorry, my cats are climbing up things. No, we don't need to talk about heparin. Okay, so I think that pretty much does it. We talked about lectins, we talked about pectin, we talked about, you know what I can mention, but I forgot to mention, that we can actually tie into this idea of the bacteria and viruses. So viruses use molecules like this, lectins, to gain attachment into our cells. The flu virus binds our sialic acids. So sialic acid carbohydrates on the outside of our cells, it recognizes it. Tamiflu, you guys are familiar with Tamiflu, maybe you know about Tamiflu. This is a medicine you can use for viruses. It inhibits a molecule called neuraminidase. I can't say it. Neuraminidase. And this essentially, by inhibiting this neuraminidase, this cuts sialic acid off. And it's necessary for the exit of the flu virus. So the flu virus needs to have this neuraminidase enzyme doing something, and that neuraminidase, I don't have all the details in front of me. I forget exactly what it does. This neuraminidase has something to do with the sialic acid, and either cleaving this molecule from it, something from it, but it inhibits that enzyme, basically blocking the flu virus from being able to exit the cells that it's trying to exit. Because most of these cells, if they don't, or most of these viruses, if they don't pop your cells by basically, causing some kind of apoptosis so that all of the internal little virus particles just get kind of spread around, then they actually exit the cell by another method, kind of called like an exocytosis, where you got a lipid bilayer cell, and then you sort of form a little bubble of another lipid bilayer, and then this goes over here and sort of merges together and then becomes part of it, and everything on the inside of this bubble ends up on the outside of the cell. And so it could have something to do with the viruses being able to gather on the inside of that little bubble. Anyways, I thought I'd mention that. Okay, so that does it for this chapter, or this lecture on carbohydrates. Definitely read the book on carbohydrates. The next lecture we do is going to be on lipids and membranes. All right, guys. Take care.